So hello everyone, welcome back to Hasvora Education. Today we'll be solving the Biology 9700 Paper 2 Variant 2, the February-March 2025 session. So the time allowed for this paper is 1 hour and 15 minutes and the total marks for this paper are 60 marks. So let's begin with the first question. I would appreciate if you can subscribe this channel and hit the notification bell so that you are updated of all the videos that are being uploaded in the future. So let's begin with the first question. So the first question says, Smilestina is a herbaceous plant. Figure 1.1 shows a part of transverse section of the roots of S. china with root hair cells visible. So these extensions are the root hair cells which you can see. These are the root hair cells, these extensions, and we can clearly see the cell structures, not the inside structures, but you can say the whole of the cells and their boundary. So name the type of the microscope that has been used to obtain the image. So since we can see only the outer boundaries like the cell membrane and cell wall of this um, in the diagram, and we cannot see the internal structures, this means that the light microscope has been used. Remember that light microscope, or you can say the electron microscope, has nothing to do with the black and white. Yes, it is the fact that the scanning electron or the electron microscope gives us the image in black and white, but light microscope can also do that. So make sure that you have a grip on these structures, like you should know that whenever the cell structures inside the cell are visible, it is an electron microscope, and whenever only the outer boundaries are visible, it means the light microscope has been used. So the answer for this will be light microscope. B part says calculate the actual length in micrometers of the root hair cell labeled in figure 1.1. Use the image length of the root hair cell along the line XY in your calculation. So first of all, what we will do is that we will measure the line XY using our scale. So I've already measured it. It is 4 centimeters. And then what we will be using the magnification which they have given us as 84, and then using the formula magnification is equal to image length, or you can say measured length, divided by actual. So magnification is 84, then the image length is 4 centimeter, we will convert into micrometer, so it becomes 4000 40,000 micrometer, 40,000 micrometer, and divided by the actual length, so actually become 40,000 divided by 84. Let's calculate the actual length. It comes out as 476 micrometer. So 476 will be our answer. Let's move on to part C, which says that root hair cells are important adaptations of the root hair cells for the uptake of water. Explain one way in which the root hair cells adapt the root hair cells for the uptake of water. So we know that the root hair cells have the extensions. So these are the extensions which actually make a significant role in the absorption of water, because they have a large surface area to the volume ratio. This means that it can increase the surface area. So you can simply write it increases the surface area for absorption and you can also write that it increases the number of aquaporin channels that are being present in the membrane. So When there are more aquaporin channels, it means that there will be more uptake of water. And you can also write that it has a significant role in reaching a large volume of water in the soil. So any of the 3 points, you can write one of them and you will get the 1 mark for it. Let's move on to part D. So part D says mineral ions are taken up by the root hair cells. Table 1.1 shows the concentration of the sodium ions and the potassium ions inside the root hair cells of the plant root and in the soil solution surrounding the root. So they have given us the concentration of the sodium ion and the potassium ion, and in the root hair cells we have a concentration of 0.35 gram per dm, and in the soil solution we have 3.34, and for the potassium we have 5.46 in the root hair cell and 0.16 in the soil solution. So with reference to table 1.1 suggest the mechanism involved, like whether it is active transport, whether it is facilitated diffusion, or whether it is simple diffusion. We have to state the mechanism, and we have to state the reasons for your suggestion, that why are we thinking of that this mechanism is involved. And when the sodium and the potassium, the transport of sodium and potassium from the soil solution into the root hair cell. Okay, so keep an eye on that. So you can see that in the soil solution we have the sodium, we have more sodium than in the root hair cell. So it means that there will be only facilitated diffusion here, and in the soil solution we have less potassium than in the root hair cell, so this means there will be active transport here. And also when you are referring to the table 1.1, make sure that you use of these figures, because it will be like you are referring to this table. Okay, so we can simply write the sodium ions will pass from soil solution to root hair cell via facilitated diffusion, as there is more concentration of the sodium ions in soil solution than in root hair cell, so it moves down the concentration gradient. Okay, and also you have to provide the figures, so we can just simply write that there is more concentration of the sodium ions in the soil solution, and in the bracket we can write 3.34 gram per dm, and then in the root hairs, and you can simply write 0.35 gram per dm. Now also write for the potassium ions, you can simply just intensify this uh, answer, so we will write while potassium ions move actively from soil solution using energy via protein carriers protein carriers as soil solution have less concentration which is 0.16, then root hair cell, which is 5.46. Hence, it moves against the concentration gradient. So this will be your answer. This is a 3-mark answer. 2 marks for the mechanism of the sodium and mechanism of the potassium, and 1 mark for the explanation of why you think that this will be the mechanism. Okay, so next part he says the region between the outer layer of the root and the endodermis is known as the cortex. Table 1.2 shows the water potential of two adjacent cells A, B in the cortex of the root. So they have given you the cortex cell A and cortex cell B. So the water potential of the cortex cell B is more negative, and the water potential of the cortex cell A is less negative. So with reference to table 1.2, explain the direction of water movement between cell A and cell B. So it is a simple question. We just have to write the mechanism that via osmosis it will move from higher to lower concentration gradient, and from higher to lower water potential gradient, and from cell A to B. So we can just write water moves from cortex cell A to B via osmosis down the water potential gradient, or you can also write from more from less negative to more negative. Then part F has a very big story. Okay, let's read it first. So some types of soils are made of small negatively charged clay particles that attract and bind. So they have given you that the clay particles are negatively charged, so that attract and bind to the positive ions such as the Fe2 plus or the iron cation. Fe2 plus that is bound to clay particles cannot be absorbed by the root hair cells. This reduces the concentration of free Fe2 plus that is available in the soil solution for absorption. Some plants that grow in soils containing a high proportion of clay particles are able to increase the concentration of free Fe2 plus in the soil solution for absorption. So now they are telling us that some plants have the capability so that they can increase the free Fe cations, like it will just dissociate the clay particles from it so that it can absorb it. So in these plants, the carbon dioxide released by the respiration of root hair cells reacts with the water in the soil solution. Okay, when this changes the pH of the soil solution, this affects the binding of the positively charged ions. So they have also linked the pH of the soil solution with the binding of the positively charged ions to the clay particles. So now they will probably asking us about how this works. So let's read it. So figure 1.2 shows the result of one investigation into the effect of the pH on the concentration of the free Fe2 plus that is available in the soil solution for the absorption by root hair cells. The soil sample analyzed in the investigation was from the soil that contained a high proportion of clay particles. Okay, so now you can clearly see that when the pH is increasing, the concentration of the free Fe2 plus is decreasing. Okay, so this means at lower pH, the Fe2 plus ions which are free increases. Okay, because when the pH is increasing, the free Fe2 plus ions are also decreasing. So with reference to figure 1.2, suggest and explain how the carbon dioxide released by the respiration of root hair cells can increase the concentration of free Fe2 plus ions for the absorption by the root hair cell. So whenever they use the word suggest, this means that you do not need to know about this procedure or the whole process that was explained to you, but you have to use your previous knowledge, or you can say the syllabus content you have read throughout the year, to answer this question. So you have seen a similar type of case in the respiration when the carbon dioxide reacts with the water to form the carbonic acid in the presence of the carbonic anhydrase, but there is no carbonic anhydrase in the soil. So then it will dissociate into the ions, and then into the H plus ions and HCO minus 1 bicarbonate ions. Um, I hope you all are taking chemistry. So whenever the H plus ions increase in the concentration in an aqueous solution, the pH becomes acidic. So that's the main idea of these questions. Now we will answer this question like the carbon dioxide released reacts with water to form carbonic acid. 1 mark for this, which dissociates to form the bicarbonate HCO minus 1O3 and H plus ion. The second mark for this. Then we have to write the H plus ion reduces the pH of soil, which decreases, you can say that decreases the attraction or the binding between clay particle and Fe2 plus ions. And you can additionally write that, let me just remove this, I just wrote it by mistake. Okay, so the HCO minus 103 ions can attract the Fe2 plus binded to clay particles. So when it attracts the Fe2 plus that is binding to the clay particles, actually it is freeing it from that attraction with the clay particles, so it becomes a free Fe2 plus cation that is being able to absorbed. So ions can attract the Fe2 plus binded to the clay particles, or you can also write that the H plus ions are binded to clay particles, because clay particles are negative, so they can bind to anything that is positive, hence releasing or setting free the Fe2 plus ions in soil solution. So this will be your 4-mark answer for this question. Let's move on. So part 2 says dissolved Fe2 plus ions is transported across the tissues of the root to the xylem. When the amount of the dissolved Fe2 plus ions absorbed by the root hair cells is greater than the amount that is needed by the plant, not all of the Fe2 plus cations that is absorbed is transported to the xylem. So this means that the plant can actually remove the excess that is being absorbed through the root hair cells. So suggest how the endodermis can reduce the amount of Fe2 plus ions reaching the xylem. So they are actually emphasizing the role of endodermis here. So we know that endodermis is made up of Casparian strip, and the Casparian strip is made up of a hydrophobic material which is called the fibrin, and it also, you can say, blocks the apoplastic conduction from the root hair cells to the xylem. So this means only symplast pathway is available, and it will limit the amount of Fe2 plus ions that are being reaching the xylem. So we can simply write the Fe2 plus ions must pass through the cell surface membrane which uh, the cell surface membrane of Casparian strip of endodermis which blocks and limits the number of Fe2 plus ions that pass through the endodermis or pass through it. It only allows simplest pathway. So because they are using the word suggest, so we can just give a, you can say, a possible reason for that. So simplest pathway, so less number of hydrophilic channels will be present to carry the Fe2 plus ions. And also you can write about the Sibrin, that it is a hydrophobic material which blocks the number of Fe2 plus cations. So this will be your answer for this question. Let's move on. So this big question has just came into end. We have just hopped into the question number 2. So figure 2.1 is an incomplete diagram of the structure of the alpha glucose molecule. Complete figure 2.1 to show the structure of an alpha glucose molecule. So you should remember that the alpha glucose has the OH group that are facing downwards. So OH here and one OH will be here. We will have an H here and H here and HN here. Then you can just simply alternate this OH group. So this will become here and H here, and then we will have an H here and OH here. Okay, so this will be a structure for alpha glucose molecule. Figure 2.2 shows the part of a glycoprotein molecule. Glycoprotein means that a type of conjugated molecule made from two different molecules that are from different categories. So it is made from a glucose, possibly a carbohydrate, and plus a protein. So this side chain you are seeing is a short carbohydrate chain, and the other one is a chain of amino acids, a protein. So what is the bond? See, it is between the amino acids, so it will be a peptide bond, and D is between the molecules of the carbohydrate chain, so it will be a glycosidic bond. So this was an easy question. Let's move on. Part 2 says many glycoproteins in the cell surface membrane are involved in the cell signaling. State a role in cell signaling of the glycoproteins in the cell surface membranes. So we have studied this already, that the glycoproteins and the glycolipids are involved in, you can say, they act as the recognition sites or the recognition markers for the neurotransmitters and the hormones. So we can just simply write they act as receptors for neurotransmitters and hormones. Okay, let's move on to C part, which says one of the proteins found in the milk is B-casein. So it is a type of protein. A molecule of B-casein consists of a single polypeptide of approximately 200 amino acids. Molecules of B-casein have a high proportion of amino acid proline and leucine. These amino acids have a hydrophobic R groups. Just remember, proline and leucine have hydrophobic R groups. Figure 2.3 compares the structure of proline with the general structure of amino acid. This shows that the proline is an unusual amino acid because it has a cyclic R group and has a nitrogen atom that is attached to only one hydrogen atom. So this means that the nitrogen is only also not available for binding via the hydrogen bonds because it is attached to the other groups. So this is the structure of proline and the amino acid. When a molecule of proline becomes the part of polypeptide, the hydrogen atom is lost from the nitrogen atom and therefore not available for the formation of hydrogen bond. Just as I said, molecules of B-casein have a relatively low proportion of the amino acids cysteine and serine. So they've also provided us with this information that it has a low concentration or you can say low proportion of cysteine and serine. Table 2.1 shows the R groups of cysteine and serine. So this is the cysteine and this is the serine. It has the OH group. It means that it can form hydrogen bonds, and the cysteine can form a disulfide bond because of its sulfur. The hydrogen will be lost and there will be a sulfur-sulfur bond, which we call the disulfide bond. Compared to the other protein molecules with a similar number of amino acids, B-casein molecule have a much less organized secondary structure. So this will be possibly because of the hydrogen bonds, because there are more hydrophobic R groups, so it will be less organized secondary structure and relatively little tertiary structure. This will be because of the less number of the serine molecules, because of the less number of the disulfide bonds, less number of the hydrogen bonds, and there is no ionic bonding they have discussed till now. So with reference to the four named amino acid, proline, leucine, serine and cysteine suggest for possible explanation for these two observations. So let me just write one thing here first. So these two are hydrophobic, this means weak attraction, and these two can form disulfide and hydrogen bond. Okay, but because they are in less number, so they will not benefit a structure more. So the possible reason for the less organized secondary structure will be, we can just simply write the high proportion of proline and leucine having hydrophobic R groups mean there will be no hydrogen bonding, so less or no alpha helix or beta pleated sheet can be made. So this can be the reason for this. And for the relatively little tertiary structure, remember that in the secondary structure we do not have the disulfide bonds, we only have the hydrogen bonds. So the relatively little structure will be because serine can make disulfide bond while leucine has the hydroxyl group or the OH group for hydrogen bonding, but since they are in less number, or you can say less proportion. The tertiary structure will be more dependent on hydrophobic interactions of proline and leucine. Also, we can write that is no there is no ionic bonding involved as well. So this means that only hydrophobic interaction will be present in the tertiary structure, making it less, you can say, organized. Then we have the third question. So figure 3.1 and the figure 3.2 are the diagrams of the transverse section of the human heart during the different stages of the cardiac cycle. The sections pass through the heart of a level that is just above the valves. Name the valve V. So you can clearly see that these valves are larger in size. These are larger in size, and you can also see that when they close, they make, you can see, three lobes, a type of three different lines. This means this will be a tricuspid valve and this will be a bicuspid valve. Okay, so this will be a bicuspid and this one will be a tricuspid valve. This means the smaller ones will be the semilunar valves that are being placed between the ventricles and between the aorta, or you can say the right ventricle and the pulmonary artery. So this will be semilunar valves. Semilunar valves. Okay, so the second part said, with reference to the chambers of the heart and the main blood vessels, describe the flow of the blood through the heart that occurs when the transverse section of the heart appears as shown in 3.1. So in 3.1 you can see that the entry ventricular valves, the tricuspid and the bicuspid, are opened and the semilunar are closed. This means that there will be a flow of blood from the atria to the ventricles, and you can say from the vena cava into the atria, but there will be no flow of the blood from the ventricles to the aorta, or from the ventricle to the pulmonary artery. So we can just simply write the blood flows from the atria to the ventricles and from vena cava and pulmonary vein to ventricles. As the entry ventricular valve, I will just write the AV valves are open, but there will be no flow of blood from atria, from the ventricle, from the ventricle to aorta or pulmonary artery, as semilunar valve is closed. Okay, so this is your answer. Identify the stage of the cardiac cycle shown in the figure 3.2. So in the figure 3.2 you can see that the semilunar valves are open. This means that the ventricle will be contracting, which we call the ventricular systole. It will contract to move the blood out of the aorta and also into the pulmonary artery, while the AV valves are closed. So the stage of the cardiac cycle is ventricular systole. The reason is the AV valves are closed to prevent backflow into the atria, and semilunar valve is opened as ventricle contracts. So this will be your answer. Let's move on to part C, which says the rate and the rhythm of the heartbeat are controlled by an area of specialized muscle tissue in the wall of the right atrium, which is called the sinoatrial node, which is, you can say, responsible for generating the waves of excitation, or what drives our heart. So describe the sequence of the event that controls the correct contraction of the ventricles during the cardiac cycle. So we just have to state the sequence of the events in the correct sequence. So we will write the SAN, the sinoatrial node, generates waves of excitation which causes the atria to contract, but due to presence of fibrous tissue they can't be transferred directly to ventricles. Hence, they are passed to the AVN antiventricular node after 0.1 second, because this ensures the AVN delay. So there is a delay between the contraction of the ventricles and the atrias. So after 0.1 second, which transfers it to the Purkinje tissue via bundle of His. So then the waves of excitation actually reaches the apex of the heart. So the waves then spread from the base upwards from the apex of heart, causing the ventriculars to contract. So this will be your answer for this question. We have just stated the sequence of events that causes the atrias to contract, and how the ventricles contract, and how the AVN delay actually happens. Okay, let's move on to question number 4. Now question number 4 says, vaccination programs are widely used to help control the spread of infectious diseases. State why some diseases are described as infectious. So we have to write the definition of what infectious actually means. So we can say they are caused by disease causing pathogen which is transmissible. It can be transferred from one individual to other person, and they are caused by an pathogen. A person was given an injection to give protection against infection diseases E. The person had not previously been infected with disease E. 26 days later, a second injection was to give protection against disease E was given again. So the concentration in the blood of the antibody specific to disease E was measured over a period of 60 days from the time of the first injection. Figure 4.1 shows the concentration of the antibody in the blood over the period of 60 days. So they have given us the whole time period when one injection was given and then the second injection was given. So these graphs look similar to what we have studied, the primary immune response when the first time the person is exposed to the pathogen, the primary immune response, this is the primary one, and this is the second immune response, and this is the concentration of the antibody is much more because of the presence of the memory cells from the first immune response. Okay, so the concentration of antibody in the blood after the second injection was higher than the concentration of the first injection. Explain why the concentration of the antibody in the blood was higher after the second injection than the first injection. So we can just simply write this. The answer will be that because of the presence of memory cells from primary immune response from the primary immune response when the person is again encountered by same pathogen, the B and T memory cells rapidly proliferate and differentiate to produce high amount or high concentration of plasma cells, and then the plasma cells can which release increased concentration, increased amount of antibodies. Okay, so the main point of the matter is the memory cells. So let's move on to the B part, which says the second person was given a different injection to the gift protection against another infectious disease F. Okay, so the person had not previously been infected with the disease F. The concentration in the blood of the antibody specific to the disease F was measured over the period of 60 days from the time to time of the injection. So figure 4.2 shows the concentration of the antibody in the blood over the period of 60 days. So we have seen that whenever a vaccination, or whenever you can say a natural encounter between the pathogen and an individual occurs, the graph of the antibody concentration is something like this. Okay, so because the it takes a while for the primary immune response to be generated, that is why it is, you can say, a type of curve. But here you can see that the antibody concentration is reaching a max point and then it is decreasing from that point. When the injection is given, the antibody concentration suddenly becomes the maximum. So this means that the individual has been given an an injection of antibodies rather than that of the pathogen for vaccination. So he is being given the injection of antibodies, ready-made antibodies. Okay, so this will be artificial you can say it will be artificial and passive because he is not producing the antibodies himself, and artificial because then you can say not a natural way is being used to give him the injection. So type of immunity will be artificial, passive immunity. Describe the features of the type of immunity resulting from the injection gives us protection against disease F. So this is a short term protection against disease, and the antibodies are destroyed. This type of immunity, this produces no immune response. Okay, so no immune response is produced, no immune response, and no memory cells. So that's why it is a short term immediate effect response against a disease. So I think this question is finished. No, it is not. C says in 2021 the number of new cases of tuberculosis were estimated to be 10.6 million. Name the bacterium that causes tuberculosis. So we can just write mycobacterium. You can also write tuberculosis, and you can also name the other variant is which is called the Mycobacterium bovis, but that is caused in the cattle. So I would just suggest that stick to the tuberculosis one because it is the main reason of the why humans get infected with tuberculosis. So TB is often treated with several different drugs at the same time. This is necessary to kill the multiple drug resistant MDR strains of the bacteria. This treatment is usually lengthy, taking 6 months or more to make sure that all the bacteria are killed. Researchers investigated how the bacteria that causes TB react to the presence of rifampicin. Rifampicin is an antibiotic that is often used in the successful treatment of the TB. So researchers combined rifampicin with the colored maker dye and added colored rifampicin to the culture of living bacteria. So they were actually testing out that what is the effect on the bacteria of the TB with rifampicin. When initially viewed under the microscope, the researcher could see that the colored rifampicin was present inside the cytoplasm of the bacterial cells. This means that the antibiotic has penetrated to the body of the bacteria into the cytoplasm. Hours later, the colored rifampicin was not visible inside the bacterial cell but was visible in the medium surrounding the bacterial cells. This means that it has been excreted out by the bacterial cells because this is how it develops this resistance. So it uses its membrane pumps to kick out the antibiotic. So the researchers concluded that rifampicin was being pumped out by the bacterial cells through the cell surface membrane, and it will be because of the pumps. Okay, so further research showed that when some drugs commonly used in the treat indigestion were added to the bacterial culture containing colored rifampicin, the colored rifampicin stayed inside the bacterial cytoplasm. So what clue is this giving you now? This says when the drugs treating indigestion, so when these are given um, to the bacterial cultures, what happens that it is unable to kick out the rifampicin on the antibiotic from the cytoplasm. So this means that it is inhibiting the membrane pumps of that bacterial cell, so it is unable to, you can say, excrete the antibiotic. So suggest and explain how the knowledge gained from this research could improve. So we can give the possible improvements of TB treatment and reduce the chance of rifampicin resistant bacteria developing. So it is worth 4 marks. So let's write this question now. So first of all, we can just write that the drugs treating indigestion could be given to people with rifampicin, so it effectively remains inside, and you can say effectively kills the bacteria, or you can say it remains inside the bacteria. So the indigestion drug inhibits the membrane pumps, that is actually, you can say, excreting the antibiotic membrane pumps in cell surface membrane of the of bacterial cell, and it will also help, it will also help build up concentration of rifampicin inside bacterial cell, that is lethal to it, so it can effectively kill it. Okay, lethal to it. This reduces the time span and cost of TB treatment, and you can also write it and you can link it with the time. So less time reduces the likelihood of bacterial genome, you can say, it is developing, from prevent the likely reduces the likelihood of bacterial genome developing resistance against rifampicin. Okay, so rifampicin, so this will be your answer for this question. Let's move on to question number 5, which says the eukaryotic cells are prokaryotic, and the eukaryotic cells and the prokaryotic cells contain the DNA. Complete the passage about the DNA in the eukaryotic cells and the prokaryotic cells using the most appropriate terms. In the eukaryotic cells, the DNA is located mainly in the chromosome of the nucleus. Chromosomal DNA is associated with the proteins called histones. You should have studied this. In the humans, there are 8 histones around which the chromosomes are being, you can say, rounded off, so that they can be accommodated in the small nucleus, because you know that the chromosomes are very longer in length. So in the eukaryotes, they are being associated with the proteins called the histones. In the humans, there are 8 histones, and you can say in the fungi, they are just, you can say, helical, uh, you can say they are the helical chromosome around the histones. And in the, you can say, the in the other species, it is just, you can say, linearly arranged around the histones. So the two other eukaryotic cell structures that contain DNA are mitochondria, and the other one will be chloroplast, because it is inside the eukaryotic cells, the plant cell. Okay, in the eukaryotic cell, for example, you can write bacteria, and you can also name any other prokaryote. Okay, so the DNA is found in the cytoplasm. Okay, it is just, we have just studied that the prokaryotic cells, they are, you can say, they are non-membranous. So the plasmid, or you can say the DNA, it is just dispersed in the cytoplasm, and it is usually circular. So they are talking about the plasmid. It is also in the cytoplasm. So figure 5.1 shows the transcription of the first 6 nucleotides of a gene by the enzyme RNA polymerase. The bases of the first 6 nucleotides on the DNA strand X are shown, but the bases on the template DNA strand are not shown. So they have given you that this one is the DNA strand X, and this is the template template strand means that this is the one that is being transcribed into an mRNA or the primary transcript. So this one will be your primary transcript. Okay, so uh, this whole structure is called a DNA fork, because you can see that it is forming a shape of a fork. And this is also called, I've just forgotten the name, so let's just move on. So this is just the name. Okay, I'm just giving you an additional information. So state and term used to describe the non-template DNA strand labeled X. So the non-template strand or non-transcribed strand, you can also write the non-transcribed strand. It means that this one is not being transcribed. And the other thing that you can write about this strand is that it is also called the sense strand. Why is it called the sense strand? Because we know that the DNA, the two strands of the DNA, when they unwind, they have the complementary base structure. Okay, so for example, if this one is A, this one will be T, and if this one is T, this one will be A, and the same way this will be C, this one will be G, and this one will be then T, and then A again. So these are complementary to each other. So whenever this mRNA is being synthesized, the structure of this is complementary to the mRNA. Okay, so the mRNA sequence and this sequence actually matches, but instead of thymine there is the uracil. So this means the sequence of the non-transcribed strand and the mRNA transcript that is being synthesized from the transcribed strand, the sequence matches. So that is why it is called a sense strand. But you can also write the non-transcribed strand. Okay, the non-transcribed is a better, you can say, alternative to it. Non-transcribed strand. Name the RNA strand formed during the transcription of a eukaryote gene. So they have made no reference of the splicing, or you can say the post transcriptional modification. So you are just going to be writing this as, you can say, pre RNA, or you can say the primary transcript. You can also write the primary transcript because it is just freshly made from this one of the strand. So this is not an mRNA. Okay, this is a primary transcript. Now it has to undergo all of the splicing, the polyanidation, the 5' capping, so then it will become the mRNA. Right now it is a primary transcript. So make sure that you are very specific to what you answer. Complete table 5.1 to show the letters of the 6 bases indicated on the figure 5.1 by the numbers 1 to 6. So I just told you that the sequence of this will match the sequence of the mRNA or the primary transcript, but instead of the T there will be uracil. So it will become A, then we will have U, G, C, and then we will have A, and then again U. So AUG and CAU. AUG and CAU. So I hope you remember that this is the methionine amino acid. This is the codon for methionine. So RNA polymerase is composed of several polypeptides that move together and change the shape as the enzyme performs its function. The death cap mushroom, the Amanita phyllides, produces a toxin called alpha-amanitin, which binds to the RNA polymerase at a site other than the active site. So whenever they talk about a site other than the active site, this means an allosteric site, and this means that it is a non-competitive inhibitor. Alpha-amanitin reduces the activity of RNA polymerase. Use the information provided to suggest how alpha-amanitin reduces the activity of RNA polymerase. So it is binding to an allosteric site. It is a non-competitive inhibitor. It will change the shape of the 3D structure of the RNA polymerase, which is also an enzyme, and it will reduce the you can say the enzyme substrate complex that is being made between the RNA polymerase and the nucleotide. It will prevent the nucleotide from binding to the RNA polymerase, and hence the activity will be reduced. So we can just write that alpha amanitin is the non competitive inhibitor of RNA polymerase. It changes, or you can say it alters the 3D structure of RNA polymerase and prevents nucleotide from binding to the enzyme. Hence, no enzyme can bind to substrate to form ES complex, and activity of RNA polymerase is reduced. Okay, so this will be your answer. Let's move on to question number 6. I hope this is the last question. Figure 6.1 shows the photomicrograph of the individual cells from the root tip of an onion, AMSP, at the different times in the mitotic cell cycle. So first of all, just let's have a look at the you can say at the different cell structures. So in the B, we can say that it is you can say a condensed form of the nucleotide, a not non-condensed form of the nuclear of the chromosomes. So it will be, you can say, the prophase. This one will be a prophase. In the diagram that is below the prophase, in E, you can just simply say that these chromosomes are now coiled enough. So this one will also be the prophase. This is just the ongoings in the prophase. Okay, now F, you can say that the chromosomes are being arranged around the equator. So this one will be your metaphase. This one will be your metaphase. And then we can say that in the D, there is anaphase, because the sister chromatids are being separated on the and they are moving on the opposite poles. In the A, you can just simply say that the now they are just being in the process of decondensing, so it will be telophase, or you can say at the end of the anaphase simply. And in the C, you will just say that it will be you can say after somewhere after the telophase. Okay, because then it is decondensing. Okay, and it is forming, you can say now after this it will form the nucleus again, the nuclear envelope again. So this one will be, these will be all your structures. Place the letters representing the individual cells in the correct sequence of the mitotic cell cycle. The first letter has already been filled in. So B, they have said that it is the first place. Then we will say that it is E, because then it is condensing. Then after that E, we will have the F, because it will be the metaphase. Then after that we will have the D, because then it is just moving to the opposite poles. At A, it is slightly you can say decondensed, and the last stage will be C, after which it will form the nuclei and the nucleus and the nuclear envelope, and all the structures will be regained. Cell A in the figure 6.1 is the one of the main stages of the mitosis. Describe the events that occur during the main stage of the mitosis. So A, we said that it will be, let's have a look, I will show it to you. So it is telophase. Okay, so it is telophase. We are talking about the telophase. So what will happen? Describe the events. So the nuclear envelope reforms, and you can also write, and the nucleus reappears, and you can say reforms. You can also write the spindle fiber breaks or disintegrates. The spindle fibers disintegrates. Okay, so this will be your answer. Okay, you can write a nuclear envelope about, you can write about the nuclear envelope, about the nucleus, about the spindle fibers, and one more thing that I think I'm forgetting. So what will it be? And also you can also write about the chromosomes. The chromosomes decondenses. Okay, and then it will decondense um, to become less coiled structures. So then we have the B part, which says complete table 6.1 by stating the term that matches each of the descriptions. The region of the DNA will represent repeated nucleotides. So sequences are located at the end of the chromosome. So this will be your telomere, because we know that the non-coding sequence of nucleotide at the end of the chromosomes that are being used, you can say, they prevent the important information on the DNA to be lost. As in the DNA replication, we have studied that the ends of the chromosomes are of the chromosomes are being, you can say, lost after each replication. So the telomere are the for this purpose. So organizes microtubule to form the spindle in the animal cells. So they are the centrioles. Point of attachment between the two sister chromatids. So it will be uh, your centromere, because it is the point, it is the you can say, let me just write it again. Okay, so centromere, so centromere is the point. This is the centromere. Just give me a second so I can draw this chromosome. For example, this is the chromosome. So this this middle part, which I have just highlighted a bit, this is your centromere, to which both of the two sister chromatids join. And in the case if they had said that to which points do at the chromatid do they join, then we could have written kinetochore in the centromere. Okay, so you have to be very specific and you have to be very vigilant when you are reading the question, that what the examiner is asking us about. And this paper was actually very lengthy. This was of 24 pages. Okay, so but it was, I think it was a very relatively easy paper. So we have completed this paper in about an hour, I think. So it's about an hour and 8 minutes. So if you like this video, do share and like and subscribe this channel, and I'll be meeting you in another video. Till then, stay care, stay safe, and goodbye.